Have you heard about the Archaea? Despite this group being one of the 3 domains of life, surprisingly, a lot of people barely know about them. But the Archaea are really incredible. There are organisms in this domain that can survive geothermal hot springs, acidic environments, and even super salty lakes, basically places where other things just can't survive. For a long time, the Archaea were thought to be just another type of bacteria because on the surface they look pretty similar. Actually, the old name for this group was the Archaea bacteria. But once researchers started digging in with genetic tools, we found that they are completely different. In fact, they're so unique that they don't fit in with either the bacteria or the eukaryotes. That's why in 1977, Carl Woese proposed the 3-domain system, which puts Archaea in their own separate domain alongside the bacteria and the eukarya. We're going to learn about some of the really cool, unique characteristics that set the Archaea apart from those domains of life, but first, let's start by talking about the similarities. Microorganisms in the domain of both bacteria and Archaea have 70 S ribosomes for protein synthesis, and they usually carry their DNA in a single circular chromosome. They're also about the same size, much smaller than the eukaryotic cells, generally speaking. And if you look on the outside, you'll notice features like flagella-like structures and pili that serve similar functions to those in bacteria, even though the details of how they're built are often quite different. Archaea also share some surprising features with eukaryotes. For instance, the way they replicate their DNA and transcribe genes look much more like what happens in eukaryotic cells than in bacterial cells. They even use histone-like proteins to help package and organize their DNA, which is a hallmark of the eukaryotes. So while Archaea may look like bacteria on the surface, at the molecular level, they actually have a lot in common with us. You can see that Archaea share commonalities with both bacteria and eukarya. In fact, this tree of life that shows the 3 domains isn't really showing the full story. Endosymbiotic theory states that the entire branch eukarya may actually be a result of some primordial Archaea-like cell engulfing bacterial cells, which evolved to be intracellular symbionts. What evidence do we have of this? With growing research, there's lots of clues, but among the most convincing are those that come about when you take a look at organelles like the mitochondria or the chloroplast. Mitochondria not only contain their own DNA, which is small and circular, just like bacterial chromosomes, but they even have their own ribosomes, which are 70S like bacteria and Archaea, rather than the 80S ribosomes of eukaryotic cells. They also have a double membrane, which draws similarities to double-membraned gram-negative bacteria alive today. Add to the evidence that the mitochondria divide independently of the eukaryotic cell, using a process that looks a heck of a lot like binary fission, and mitochondrial DNA is genetically more similar to alpha proteobacteria than anything else. It tells a story that some ancestral form of Archaea and bacteria formed a symbiotic relationship, an event which likely happened more than once, to eventually evolve into the eukaryotic cells we see today. Too cool. But how are the Archaea different from both bacteria and eukarya? So what are some structural characteristics that set the Archaea apart from the other 2 domains of life? Let's start with the cell membrane. Instead of having straight-chain fatty acids typical of bacteria and eukaryotes, archaeal lipids contain isoprenoid chains, which are often branched. These branched chains increase membrane rigidity and reduce permeability, allowing the cell to maintain its internal environment even in environments like high salt lakes or hydrothermal vents. Archaea also have ether linkages connecting glycerol to their lipid chains, instead of the ester bonds that you find in other cells. Ether bonds are much tougher. They resist heat, acid, and chemical breakdown, so the membrane stays intact even in hot springs or acidic pools. The glycerol backbone in archaeal lipids has an L-isomeric form, unlike the D form in bacteria and eukaryotes. This might sound small, but it changes how the lipids pack together, making the membrane more stable under harsh chemical or thermal conditions. And in some archaea, lipid molecules span the membrane to form a single monolayer rather than a bilayer. This structure is exceptionally stable at very high temperatures, preventing the membrane from falling apart. This makes me think about the Archaea that survive in geothermal hot springs. You're probably seeing a theme here. Now let's examine the cell wall and outer layers. Archaea display a wide variety of cell wall types, always adapted for the environment of the organism.
Some archaea even lack a cell wall altogether. Archaea don't have peptidoglycan like most bacteria, but some make pseudomurin. Instead of NAM, it contains N-acetylallosaminuronic acid, NAT, linked to NAG subunits, with peptide interbridges to increase strength. Others produce methanochondroitin, a polymer like the connective tissue molecule chondroitin in vertebrates. Some species have a protein sheath, a lattice similar to an S-layer, that can enclose entire chains of cells rather than just individual cells. Now let's talk the S-layer. While it's not universal, a large number of archaea do have a proteinaceous S-layer that is considered to be part of the cell wall itself. The archaeal S-layer can be made of either protein or glycoprotein, often anchored into the plasma membrane of the cell. The proteins form a two-dimensional crystalline array with a smooth outer surface. The S-layer is a protective protein coat that helps archaea survive in extreme conditions. It gives the cell shape and strength, shields against heat, salt, or acid, and can help cells stick to surfaces or each other. Think of it as a flexible molecular armor that keeps the cell safe in harsh environments. Now, external structures. Archaeella is the archaea version of flagella. It has a similar function to the flagella found in bacteria. They allow cells motility, but they're built differently from bacterial flagella. Notably, they're thinner with multiple types of flagellin protein. Interestingly, they've even been shown to aid archaea in the attachment to surfaces and other cells. The hamus, or hami for plural, is another structure unique to just archaea. This structure is a long, helical filament with 3 hooks at the end. Hami help cells stick to surfaces and each other, promoting the formation of stable microbial communities. Lastly, there's the Archaea unique cannulae. These are hollow tube-like structures that connect cells after division, forming dense networks. These structures have been observed in marine archaeal strains, such as Pyrodictium, which thrive in high-temperature environments. The formation of these networks may serve to anchor communities to each other in surfaces, and potentially aid in nutrient exchange, which is really crucial for survival in challenging conditions.
So now you know how the archaea are similar to both the bacteria and the eukarya, alongside some key characteristics that make them unique. We also saw many examples that clearly demonstrate the link between structure and function when it comes to archaeal cell structure. And it's thanks to some of these really unique archaeal structures, like for example the modified cell membrane, that we find archaea where no other life exists.